Hello everyone, welcome back to Infinity Learn by Shri Chaitanya. My name is Kiran Chaudhary and I am your botany faculty at Infinity Learn. My lovely students, today we are going to discuss all the important PYQs NTA has already asked in the previous history of NEET exam. I will be discussing questions from 2013 to 2021 NEET exam. So let's get started, and before starting I would love to say that if you are new to this channel, do consider subscribing to Infinity Learn NEET channel for more such videos and content in future. Next, this is the very first question, students, and I will also appreciate if you will participate in the session by trying to give the answers. So it doesn't matter whether you know the right answer or not. What really matters is what I expect you at max in this session is give it a try. If your answer is correct, perfect. If it is not, I assure you that you are not going to make the same mistake again in your coming NEET 2022 exam. But for that to happen, at least you have to give it a try. So if you see this question, kids, this question was asked in NEET 2020, right? So which of the following statement is correct? You are supposed to find out which of the following statement is correct. So give it a try, my lovely kids. What do you think is the right answer? Come on, give it a try. Give it a try. So if you see, let me read out the statements for you. You were supposed to find out the correct statement. Adenine pairs with thymine through 2 hydrogen bonds. So remember, students, DNA is actually a double-stranded molecule, and in DNA, if you notice, there are 4 types of nitrogen bases that are found: A, T, G, C, and it's a double-stranded molecule. I'm so sorry for this one. It's a double-stranded molecule. That simply means A is going to be bonded with thymine with the help of 2 hydrogen bond, and guanine will be bonded with cytosine with the help of 3 hydrogen bonds, and that's what is asked in this. Adenine pairs with thymine through 2 hydrogen bonds. That's absolutely correct, my lovely kids, right? Let's see the next question. If the distance between 2 consecutive base pairs is 0.34 nanometer and the total number of base pairs of a DNA double helix in a typical mammalian cell is 6.6 into 10 raised to power 9 base pair, then the length of the DNA is approximately. So see, my students, this question, the answer to this is directly given in form of fact or data in your NCERT, where packaging of DNA or double helical DNA is mentioned. This answer is directly given and the length is actually 2.2 meter, right? So this is something you got to remember. But if you ask me how to calculate how we get to this 2.2 meter, the answer is also, the method is also very simple. Total length of DNA equals to total number of base pairs into distance between consecutive base pairs. So what are consecutive base pairs? Just one next to each other. So like this is base pair 1 and this is base pair 2. So just the 1 and 2 you can say, right? So the distance between 2 consecutive base pair multiplied by the total number of base pairs present in the entire length of the DNA, you can get the total length of the DNA. So according to this, what is the total number of base pairs? 6.6 into 10 raised to the power 9. And what is the distance between 2 consecutive base pairs? It is 0.34. So into 0.34 nanometer. But if you're going to multiply it directly, you will get answer. Hello Savita, welcome to the class. So if you directly multiply this, you will get the answer in nanometers. So to get answer in meter, you have to multiply it by -9, because 1 nanometer equals to 10 raised to power -9 meter. So in this way, you will get answer as 2.2 meter. Right? It's 2.2, okay? Do not get confused. It's 2.2. So I hope now nobody will be confused and you will have a clear idea how to get to this answer, but at the end you got to remember the data as well, right? Please do not invest your time in calculating this much. Next question. Okay, so actually I placed this diagram so that I can explain you the meaning of consecutive. So consecutive base pairs means this is base pair 1 and this is base pair 2. So distance between these two base pair is the consecutive base pair. Distance between two consecutive base pairs. Next question. This was asked in NEET 2019, my kids. Purines found both in DNA and RNA are. Come on Savita, give it a try. Hello my dear students, may I know your name? Bibliophile boy, that's a nice name, quite interesting. If you don't mind, may I know your name so that I can call out your name properly. Okay, so purines found both in DNA and RNA are. So let me tell you my dear students, there are two categories of nitrogen bases. There are two categories of nitrogen bases. First one is purine, they are double cyclic. Another one is pyrimidine, they are single cyclic. Okay, so if I talk about purine, my kids, purines are A and G, adenine and guanine, and pyrimidine are actually three: cytosine, uracil and thymine. So if I talk about purine, both adenine and guanine are present in DNA as well as RNA, both. So according to this you get your answer, purine found in both DNA and RNA are adenine and guanine. But let me talk about this also my dear students. C and T, cytosine and thymine are present in DNA, right? And C and U, cytosine and uracil are present in RNA. So that means thymine is exclusive for DNA and uracil is exclusive for RNA. Is that clear? Okay, Karthik. Hello Karthik, very warm welcome to this class. Let's do this now. Next question. This was asked in NEET 2017. The association of histone H1 with a nucleosome indicates what? Any guesses over this, Karthik and Savita? Any guesses over this, Karthik and Savita? What do you think is the right answer? Come on, give it a try. And remember my lovely kids, trying is the maximum I expect you in this class. Trying is the maximum I expect you in this class. So Savita thinks the answer is B. And yes, Savita, you're absolutely correct over this. The answer is DNA is condensed into a chromatin fiber. So what you want to remember from here is, my dear students, when it comes to a eukaryotic cell, packaging of DNA in a eukaryotic cell take place with the help of histone protein. What are histones? They are basic proteins, right? And histones are actually of 5 types: H1, H2A, H2B, H3 and H4, right? So if I talk about histone, histones are basic proteins. Let us revise a little about histones. They are basic proteins. And how many types? There are 5 types. 5 types. So it is H1, H2A, H2B, H3 and H4. So H2A, H2B, H3 and H4, they will occur in a pair. All the 4 will occur in a pair. As a result, how many molecules of the histones will be? 8, because each of these 4 will be in a pair. So they will form a histone octamer. They will form a histone octamer, also known as new body, right? It will form an histone octamer. Now around this histone octamer, which is a basic protein, positively charged protein, the negatively charged DNA will wrap around, right? And this will do a formation of a nucleosome. So let me show it like this. So this is your histone octamer. This is your histone octamer. And this one is your negatively charged DNA. So this is, let's say, your negatively charged DNA, right? So this is DNA. So what is the role of H1? If you ask me, ma'am, what is the role of H1? So let me tell you, my lovely kids, you will find the H1 histone over here, and this is the histone octamer. It is made up of H2A, H2B, H3 and H4 all in pair, right? So this is how your nucleosome look like. This is how your nucleosome look like. So what is a nucleosome? Where you have a histone octamer and a negatively charged DNA is wrapped around it. So this whole structure where the incoming DNA and outgoing DNA will be sealed by H1 histone, which is the question. The question is about H1 histone. Yeah, that's perfect. This is how your nucleosome look like. So what you need to remember is whenever you see H1 histone, that means DNA is actually packed in form of nucleosome, which help in forming chromatin fiber. So this is correct. The DNA is condensed into a chromatin fiber when you see H1 histone, the presence of H1 histone. So listen this, my dear students, DNA in a non-dividing cell, I repeat myself, DNA in a non-dividing cell is present in packaged form. So DNA that you find in a non-dividing cell is actually in form of chromatin, and chromatin itself is a packaged form of DNA where double helical DNA is packaged with the help of nucleosome, where nucleosome is made up by wrapping a negatively charged DNA around a positively charged histone octamer. So these are the nucleosomes, helps in packaging, condensely packaging DNA in form of chromatin. Right, and that is why you can see chromatin as a repeating, that is why you can see nucleosome in chromatin as a repeating unit, as bead on the string structure under an electron microscope. I hope this much is clear for everyone. Is this clear for everyone? Savita, Karthik, any doubts here? I can't see any activity. Is this much clear? Any doubts? This question was asked in NEET 2015, although the exam got cancelled, but that doesn't matter. Question is a question. So in a sea urchin DNA, which is a double stranded, 17% of the base is shown to be cytosine. So first of all, you need to understand is it is DNA and it is double stranded, right? So according to the salient features, right, that are discussed in the DNA under the subtopic structure of a polynucleotide chain, under the salient feature, my dear students, salient feature of the DNA as explained by the double helical model proposed by Watson and Crick, they say that in a DNA, A always pairs with T and G always pairs with C. So in this question, the C means cytosine is 17%. Right, and we already know that C will always pairs with G, that means total percentages of C and G should be equal. Since C is 17, G should, also should be 17%. So total amount, if I talk about my lovely kids, total of G plus C equals to 34%. Now total amount of nitrogen bases, if I say A plus T and A plus T plus G plus C. So total amount will be 100%. Total is 100%. Total nitrogen base is 100%. You already know G plus C is 34%. G plus C is 34%. So that means A plus T equals to 100 minus 34. So A plus T, adenine plus thymine, equals to 100 minus 34, that is 66%. And we already know by this fact that the total number of adenine will be equal to total number of thymine, and where I have already explained you, proved you, derived that adenine plus thymine equals to 66%. That gives an idea because A equals to T, hence you can say percentage of A equals to percentage of T equals to 33%, correct? Right? So the answer is G 17%, A and T 33%, clear? Yeah, that's absolutely correct, Savita. Next question. This was asked in NEET 2015. Which of the following is not applicable to RNA? Come on, give it a try, my kids. Which of the following is not applicable to RNA? Just for the hint, I can tell you that RNA is a single-stranded molecule. 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 Any guesses, Savita? My dear, any guesses over this? Okay, then let me help you out. So, RNA definitely have heterocyclic nitrogen bases like adenine, guanine, cytosine, uracil, all these are heterocyclic. Chargaff rule, yes, that's the point. Chargaff rule is not applicable on RNA because Chargaff rules are applicable, they are applicable only on double stranded molecules. They are applicable only on double stranded molecules. Complementary base pairing rule is applicable on RNA. At 5' end of RNA there is phosphoryl, and at 3' there is hydroxyl. So only Chargaff rule is not applicable because RNA is single stranded. Excellent, Savita, excellent. Karthik. Let's see the next question. This was asked in fact twice, my dear students, in NEET 2021, that is the latest NEET exam, and also in 2013, means the first and the latest. So that means this is very famous question. The diagram shows an important concept in the genetic implications of DNA. Fill in the blanks from A to C. So what do you think is from A to C? This question is from Central Dogma of Molecular Biology. This question is from Central Dogma of Molecular Biology. So it's very simple. Any guesses, Savita and Karthik? Would you like to try it once? Come on. Give it a try, my lovely kids. Come on, give it a try. Yes? Okay. So from DNA to mRNA, the process is transcription. From mRNA to protein, the process is translation, and then this scheme of Central Dogma was proposed by Francis Crick. Hence, the answer is A. Excellent. Hello, Nikhil. Very warm welcome to the class, and very good. Your answer is definitely correct over here. Next question. This was asked in NEET 2018. The experimental proof for semi-conservative replication of DNA was first shown in. So see students, DNA actually replicates semi-conservatively. What do I mean by that? That simply means, students, that whenever a parental DNA is going to replicate to form two daughter DNA molecule, each of the two daughter molecule DNA will have one parental strand and one newly synthesized strand. This is known as semi-conservative mode. Let's say this is a parental, DNA molecule. When this is going to replicate, it will form two daughter DNA molecules, right? So each of the parental strand will have one newly synthesized strand just in front of it. So these both are the daughter DNA strands. These are the daughter DNA molecules, and in these daughter DNA molecules, one strand is the yellow one is the parental strand and blue is the newly synthesized. So hence we say DNA replicates semi-conservatively. Why? Because one strand is parental and another one is new, right? And it was first shown by Messelson and Stahl on E. coli bacteria. That's perfectly correct. E. coli. Later on it was proved over higher organisms too, but first E. coli. So, and that's the simple experiment you can see, my dear students. It's given in NCERT, right? Messelson-Stahl's experiment. So this question was asked from this experiment. I hope you can correlate now. Next question is from 2018. Select the correct match. Come on, try this one, kids. Nikhil, Savita, Karthik, come on, try this one. Try this, my lovely kids. Nikhil says A. That's absolutely correct, Nikhil. The answer is ribozyme. Ribozyme is actually an enzyme which is chemically an rRNA molecule. It's a nucleotide. It's a nucleic acid. Generally enzymes are proteinaceous in nature, but there are some exceptions like ribozymes which are nucleic acid in nature. So ribozyme is nucleic acid. To be more precise, I can give some examples like 23SR-RNA. It acts as an enzyme during peptide formation in a prokaryote or a bacterial cell. That's absolutely correct. Let's see the other option as well. Yeah, it is many a time, Nikhil, that's absolutely correct. This is many a times asked in a NEET exam. Generally, NTA put this question as one of the options in multiple statement type of questions. So it is quite frequently asked. You can easily expect this question may not be directly in, like the entire question may not be dedicated to it, but you can definitely see it as one of the option like in match the correct or in multiple statement type of question. F2 into recessive parent is dihybrid cross? No, it is actually a test cross. Next is T.H. Morgan transduction. No, dear students, Zinder and Lederberg are the one who discovered transduction and not the Morgan. Morgan is basically concerned with linkage. And G. Mendel transformation? No, it is actually Griffith who worked on transformation and gave the transformation principle, and Mendel is regarded as the father of genetics, right? I hope you get you are in a comfortable zone to understand which one is right and why the others are wrong. Let's see the next question. This was asked in NEET 2017. The final proof for DNA as the genetic material came from the experiments of. Any guesses over this? Come on, give it a try, my kids. From where does the final proof that DNA is the genetic material came? So, before the conclusion that DNA is a genetic material, there was a race between DNA, RNA and protein. But it was the experiment of Hershey and Chase which gave the unequivocal proof. Without any doubt, DNA became or was accepted by the community of the scientists that yes, it is the genetic material. They worked on E. coli and bacteriophages. Excellent. Let's see the next question. This was asked in phase 2, 2016. Taylor conducted the experiments to prove semi-conservative mode of chromosome replication on. Any guesses over this? So, semi-conservative mode of DNA replication was first of all, for the first time, was explained or was proved over E. coli, but later on that even DNA present in chromosome also replicates semi-conservatively was proved by Taylor and his colleagues, and they did an experiment on Vicia faba, which is faba beans in common. Excellent. Next question. This was asked again in NEET 2016 phase 2. A molecule that can act as a genetic material must fulfill the traits given below except. Any guesses? So, which of the following? So, out of here, my dear students, out of the 4 options, you are supposed to find out which of the following is not a property of a genetic material. Any guesses over this, Savita and Nikhil and Karthik? Any guesses over this? Come on, my kids. Okay, let me help you then. Genetic molecule, a molecule that wants to act as a genetic material should be able to express itself in form of Mendelian characters. That means should be able to express or should be able to produce a type of a protein which is going to influence a reaction which is going to bring a character. So, that is absolutely correct. And equally followed by RNA and DNA. It should be able to generate its replica, that's correct, equally followed by RNA and DNA. It should be unstable structurally and chemically, and that's wrong, and that is what I will say, this is the exception. So, this is incorrect. So, the genetic, the chemical that wants to act as a genetic material should be stable structurally as well as chemically, and in fact this is the only point or property of a genetic material where DNA won over RNA. So, RNA is structurally and chemically reactive, more reactive in comparison to DNA, and DNA is more stable chemically and structurally. So that is in fact incorrect one. It should be able to provide the scope for slow changes that are required for evolution. This is equally valid for DNA as well as RNA. That's right. Next question. This was asked in phase 2, 2014. Transformation was discovered by. Come on, this is like nothing simpler than this question can be asked from this particular chapter. What do you think? Who discovered transformation? Ma'am, DNA is stable is proposed in Griffith experiment. Yeah, definitely. Otherwise, how a DNA from a heat-killed S strain could have transformed a live R bacteria? Definitely it was. Next is transformation was discovered by Griffith. Definitely it was discovered by Griffith. So Griffith did his experiment over Streptococcus pneumoniae bacteria, my dear students, where he saw that a chemical, he wasn't sure whether the chemical is DNA, RNA or protein. He named it randomly as transforming principle to that chemical that was transferred from a heat-killed S strain bacteria to a live R bacteria, because of which the live R bacteria got transformed from R to S and was able to produce a mucus coat over it and became virulent by causing pneumonia in the rat. Is that okay? Is that satisfying for you? Nikhil, Karthik, Savita? That's absolutely correct. It's Streptococcus pneumoniae bacteria. Okay, then. So that's what I wanted to explain in this question. Next question. Let's take the next question. During DNA replication, Okazaki fragments are used to elongate. During DNA replication, Okazaki fragments are used to elongate. Any guesses over this? Come on, my lovely kids. Any guesses over this? Give it a try at least. So during DNA replication, Okazaki fragments are used to elongate the lagging strand away from the replication fork. Lagging strand away from the replication fork. So these are the lagging, this is the lagging strand, this is the lagging strand, and these are the Okazaki fragment. They are trying to elongate the lagging strand away from the replication fork. Correct? Yeah. Next question. This was asked in NEET 2021, very latest. Identify, I personally like this question. Why do I like so? Because in this single question, the entire transcription topic is actually revised. I mean, the entire transcription, whatever concepts you have learnt, examiner is trying to test in the single question is a statement based question. You are supposed to find out the correct statement. Let me see who can give the right answer to this one. Give it a try. Yes, so what I'll do is I will read out all the statement and then I'll give you the answer. Split gene arrangement is a characteristic of a prokaryote. That's absolutely wrong. It's of eukaryotes. Split gene arrangement means presence of introns and exons, right? Introns are the non-coding sequences, exons are the coding sequences. So presence of introns and exons in gene is known as split arrangement of the gene, and which is a characteristic of eukaryotic gene and not the prokaryotes. In capping, methyl guanosine triphosphate is added to the 3'. Definitely capping is done, tailing is done, splicing is done in a eukaryotic DNA or eukaryotic gene. So these are the steps followed during post transcription. After transcription, these are the post transcriptional processing, right? But in this, capping is added, or cap of methyl guanosine triphosphate is added to 5' end and not the 3'. At the 3' end, my dear students, a poly A tail is added. RNA polymerase bind with Rho factor to terminate the process of transcription. That is absolutely correct. So when it comes to a prokaryotic cell or when it comes to prokaryotic genes, my dear students, please remember, in a transcription process, my dear students, RNA, rho factor and sigma factor, these are the two important substances or chemicals, my dear students. They provide specificity to the RNA polymerase. So sigma factor binds to the RNA polymerase and helps the RNA polymerase to identify from where it has to start the transcription, and when it is the time to cease or stop the transcription, the RNA polymerase binds with the rho factor, which helps to terminate the transcription. So rho and sigma dependent transcription is seen in prokaryotes. That's absolutely correct, like bacteria. The coding strand in a transcription unit is copied to an mRNA. So please notice, my dear students, during transcription in a transcription unit, it is not the coding strand, it is rather the template strand. It is rather the template strand which is coded. Is this clear, my dear students? Hence only the third statement is correct. Hence only the third statement is correct. Any doubts here? Okay, excellent. Let's see the next question. This was asked in NEET 2020. Name the enzyme that facilitates the opening of DNA helix during transcription. Any guesses over this? Come on, my lovely kids. Any guesses over this? Okay, any guesses over this? So this is a little tricky question, and if you are really in very hurry state, you will miss out the most important hint, that is transcription. So if you do not read out transcription, you might understand or might relate this question with the replication, right, and give the answer helicase, which is wrong. So helicase, DNA helicase opens the helix in transcription? No, in replication. So when it's the case of transcription, it is the RNA polymerase that opens the DNA helix. So it was a tricky question. Please remember this. Excellent. Now let's see the next. Okay, so this is very famous. This was asked in latest 2021 and Odisha 2019, and let me tell you, in 2020 it wasn't asked exactly in this way, it was in sentence form, but this is the most easiest form in which you can remember this. That is why I'm using this format. So this is a very famous question, and multiple times it gets repeated. So when it comes to a prokaryotic cell, my dear students, all the prokaryotic DNA, all the types of RNAs, right, major types of RNAs are produced via or through single RNA polymerase. But when it is the case of eukaryotes, there are different RNA polymerase for different types of RNA molecules. So RNA polymerase 1 will form different types of rRNA, right? And so RNA polymerase 1 will form different types of rRNAs, RNA polymerase 2 will form hnRNA. hnRNA is the primary transcript. hnRNA will undergo processing to produce mRNA. So after capping, tailing, splicing, hnRNA will get converted into mRNA that will participate in translation, that has the codes, codons, that holds the information to produce a particular type of functional polypeptide chain or a protein. RNA polymerase 3 is going to produce tRNA. Is this clear, my kids? Is this clear? Any doubts till now? Let's see the next question. So this was asked in NEET 2018. Select the correct statement. Select the correct statement. So can you identify the correct statements, my kids? Can you identify the correct statement? Can you identify the correct statement? Nikhil, Karthik, Savita, can you identify the right statement? Okay, so Franklin Stahl coined the term linkage is wrong. It was Morgan who did so. Punnett square was developed by a British scientist. That's absolutely correct. It was developed by British geneticist Reginald C. Punnett. That's absolutely right. Spliceosomes take part in translation? No, spliceosomes are the ones that are going to cut or remove the introns from the exons. It is used in post-transcriptional processing, and it is used definitely after transcription, post-transcription. It is not used in translation. Transduction was discovered by Altman? No, by Zinder and Lederberg. So only B is correct. Next one. That's absolutely correct. Next question, NEET 2018. So this is the sequence of nucleotides. This is the sequence of nucleotide. from the coding strand of a gene, okay, which of the following corresponding sequence of the transcribed mRNA. So please remember, my dear students, in a transcription unit there is a template strand and there is a coding strand. Template strand actually participate in the transcription process, right, my dear students, over which RNA polymerase is going to produce a new RNA molecule, right. But remember, my dear students, the sequence of nucleotides that will be present in an mRNA, or you can say hnRNA also primarily. So the nucleotide sequence will be in mRNA will be identical to the nucleotide sequence that is there in the coding strand, but it will be complementary to the template strand, right? Because using the complementary base pairing rule, the RNA molecule or the sequence of nucleotides in RNA molecule will be decided. Hence, the sequence of nucleotides in mRNA is going to be complementary to the template but identical to the coding strand, but with one just difference, that wherever there is T on the coding strand, there will be U on the RNA, because RNA does not has T. Hello Ruxana, very warm welcome to this class. So absolutely correct, you all are correct, my dear students. Answer is A, because here, so this is the coding strand, so exactly the same sequence will be there in the mRNA, except where there is T, there will be U. So AGG instead of T, U, there is U, A T ki jagah U, C G C A and U. Absolutely correct. Next question. This was asked in NEET 2017. Which of the following RNAs should be the most abundant in animal cell? Any guesses, my lovely kids? Any guesses? Any guesses over this question? Very simple. It is the rRNA. So roughly 80 to 85% is rRNA. Then tRNA is 10 to 15%. tRNA is 10 to 15%. mRNA is 5%. Next question. Spliceosomes are not found in the cells of. Question was asked in 2016. The hint is very simple. Spliceosome has the function of splicing, and splicing means removal of the introns, and this arrangement of exon and introns in a gene is known as split arrangement, and split arrangement of gene is a characteristic of a eukaryote. So spliceosome will have no role in a prokaryotic cell. So can you identify which of the following is a prokaryote in this? Can you identify? Yeah, that's absolutely correct. So it's bacteria, it's a prokaryote, and in prokaryote spliceosome has nothing to do. This question, the equivalent of structural gene. This was asked in phase 2 2016. Structural gene is equivalent to cistron, which codes for a polypeptide chain. Muton is a unit of a chromosome that participates, smallest unit of a chromosome that participate in mutation. Operon is a set of genes where you have a polycistronic genes, like for example lac operon, you have ZYA polycistronic gene, a common promoter, operator, regulatory gene and its promoter, etc. Right? And recon is the smallest unit of the chromosome that participate in recombination. So the correct answer is cistron. Next question. 2016. Which of the following rRNAs act as RNA as well as ribozyme? See, the question has repeated again, right? So who is the one? It is 23S RNA in bacteria or a prokaryote that act as an enzyme ribozyme. This enzyme is not proteinaceous, it is nucleic acid in nature, and it is going to catalyze the peptide bond formation. That means during translation it will have its role. Excellent. DNA dependent RNA polymerase catalyzes the transcription on one of the strand of the DNA, which is called. This is already I have discussed. This question I have already discussed. Any guesses on this one? Come on, my lovely kids, give it a try. This is pretty simple question. This is a pretty simple question. I want everybody to try this one. Come on. Very simple question. Ruxana, Nikhil, Savita, Karthik. The answer is A, template strand. Template strand is coded. Coding strand is not coded. Coding strand is a misnomer. Coding strand is not coded. Template strand is coded. Template strand has the polarity 3:5. Coding strand has the polarity 5:3. And because RNA polymerase has the limitation of polymerizing only in the direction 5:3, hence 3:5 direction strand becomes template. Yes, please remember this. Do not make the same mistake again. So answer is template strand. Okay, so the question again in 2014 this question was asked and the answer is pretty simple. I have already discussed the template strand. The direction of RNA synthesis is 5:3, right? And the direction of reading of the template strand is 3:5. Because RNA polymerase can synthesize or polymerize only in the direction of 5:3, hence the strand having 3:5 becomes template. Yeah, that's absolutely correct, Nikhil. Ruxana, Savita. Next question. This was asked in 2019. So these are the important questions. Now we will be discussing about genetic codes, my kids. The question is, under which of the following condition will there be no change in the reading frame of the following mRNA? So this is one of the property of genetic codes, my dear students. Because genetic codes are made up of 3 nitrogen bases, hence deletion or addition of 3 consecutive nitrogen bases or multiples of 3 will not going to change the reading frame from the point where insertion or deletion is done. So hence deleting GGU, 3 consecutive base pairs from 7th, 8th or 9th position is not going to change the reading frame from where the deletion is done. But by adding 1 or 2 or deleting 1 or 2 nitrogen bases is definitely going to change the reading frame. That means from the point where deletion or insertion is done, the codons will change. But this change will not occur when the deletion or addition is in number of 3 or multiples of 3 consecutively. So first one is correct. Right, kids? Yeah. So this, which of the following feature of genetic codes allows the bacteria to produce human insulin by recombinant DNA technology? So which of the salient feature or property of genetic code enables them or enable us to obtain human insulin even from a prokaryote like a E. coli? So how come this happens? How can we use this or manipulate this in genetic engineering? Any guesses over this, kids? Come on, Ruxana, Nikhil, Savitha, Karthik. Any guesses, kids? Any guesses, my lovely kids? So the answer is, let me help you out in that case. The answer is, that's because genetic code is nearly universal. What do I mean by that? So the explanation is, for example, one genetic code, let's say UU, it codes for phenylalanine, right? Now notice the same codon, whether it is a human or an E. coli, a prokaryote, in both these, my dear students, UU will code for phenylalanine. That is why we are capable, or we can somehow manipulate the genes, the DNA of E. coli and get the human insulin produced in them or by them, right? But if you are guessing why the genetic code is not completely universal, because there are some exceptions because of which making the genetic code nearly universal. The exceptions you can find in mitochondrial DNA and sometimes in protozoans, in some protozoans, okay, but nearly it is universal. Next question, that's a pretty good question. 2017. There are 999 bases in RNA that code for a protein with 333 amino acid, and the base at the position 901 is deleted such that the length of the RNA becomes 998 bases. How many codons are altered? So in such a question, my dear students, let me help you out. Let me teach you what is the What is the approach that you should follow? Because they are asking about how many codons are altered. Let's start from here. So how many codons are there in total? So total codons before deletion. Deletion, right? Yeah. So before deletion, total number of codons before deletion. So how many nitrogen bases are there in one codon? 3. So how many nitrogen bases are there? 999. So 999 divided by 3, because one codon is made up of 3 nitrogen bases. So how many are there? 333, right? Obviously there are 3 amino acids. Oh sorry, there are 3 codons, and each codon is going to code for one amino acid. Hence there are 3 amino acids that can be coded. Now where is the deletion done? At 901 position. So let's say these are the total. This is the first nitrogen base and this is the 999 nitrogen base, and somewhere over here 901 it was deleted, right? So that means from 1 to 900, from 1 to 900 there is no change, right? So you need to find out, because remember the point where the insertion or deletion is done, after that point the alteration will be seen. So if the deletion was done at 901 position, that means the codons that are there after 901, here there will be alteration and here there will be no alteration. No alteration means no change. There will be no change or no alteration. I guess you might get confused by what I have written over here. So here will be no change. And here there will be change. So let's find out how many codons are there before 901 which will not show any kind of alteration. You just have to minus that much number from 333 to get the number of codons that will get altered. So how many bases are there from 1 to 900? 900 bases. Divided by 3, that means 300. That means there are total 300 codons before this point where deletion was done. So these 300 codons will not get altered. These 300 codons do not change. So how many will change? How many will get altered? Total 333 minus 300. So how many will change? So number of altered codons will be 333 minus these 300. So how many? Pretty simple, 33 codons will change from the point where insertion was done. That's absolutely correct. Next question. Which of the following is a starter codon? Starter codon, that is going to start the process, initiate the process of translation. Which of the following acts as a start codon? Come on, this is a pretty simple question. I expect everybody to answer this. Come on, my kids. Roxana, Nikhil, AUG, that's absolutely correct. AUG codes for methionine also. Please remember it has a dual function, and UAA, UAG and UGA are the stop codons. They are going to cease the translation process. Next question. This was asked in 2013 Karnataka. Which of the following is a property of, which of the following is not a property of genetic code? Which of the following is not a property of genetic code? Any guesses? Any guesses over this? Come on. Give it a try, my lovely kids. Trying is maximum that I expect you to do in this class. Yes? Ruxana, Nikhil, Savita, Karthik, give it a try, my kids. That's absolutely correct. Genetic code is not ambiguous. What does ambiguity means? Ambiguity of codons. Ambiguity of codons means what? So let's say this is a genetic code GC. This is a genetic code 1. This genetic code 1 codes for one particular amino acid, right? So this is known as what? This is known as non-ambiguous nature. But if this genetic code simultaneously code for another type of amino acid, then this is known as ambiguity. So what does ambiguity means? A single genetic code coding more than one amino acid at a time is known as ambiguity, and this is not something that is seen in genetic code. Genetic codes are not ambiguous, but genetic codes are degenerate. What do I mean by degeneracy? Degeneracy simply means, so these are the two important terms, my dear students, where students generally get confused, ambiguity and degeneracy. So genetic code is not ambiguous, but it is degenerate. Degenerate means the amino acid, let's say amino acid 1, or first type of amino acid also you can say, any X type of amino acid, this particular amino acid can be coded by genetic code 1, genetic code 2, genetic code 3, genetic code 4. So there can be multiple genetic codes, different types of genetic code that can code for a single type of an amino acid. This is known as degeneracy. But here if you notice, at a time one genetic code is coding one amino acid. No two amino acids are coded by one genetic code. So whenever two amino acids are going to be coded by one so sorry for the slip of tongue, whenever more than two amino acids will be coded by one genetic code at a time, this is ambiguity not seen in codons. But genetic codes are degenerate, that means many different types of genetic codes can code for one type of amino acid. For example, valine. Hello Shekhar. Okay, that's so nice. Thank you for sharing with me. So what I want to mention over here is, my dear students, for example, valine is a type of an amino acid. It is coded by 4 different types of genetic codes. Similarly, arginine, leucine, serine, they are coded by 6 different types of genetic code, proving that the genetic code is actually degenerate but not ambiguous. Next question. This was asked in NEET 2020. The first phase of translation is, the first phase, what is the first step? What is the first phase of translation? Translation is the process phenomenon where polypeptide chain proteins are synthesized. So can you guess which of the following is the first phase, my lovely kids? Yeah, that's absolutely correct. Amino acylation of tRNA or charging of tRNA. Charging of tRNA. So what is charging of tRNA? Charging of tRNA or amino acylation of tRNA simply means, my lovely kids, that an activated amino acid, which is bonded with an ATP, is an activated amino acid. So during amino acylation of a tRNA, charging of a tRNA, activated amino acid will be covalently bonded to its cognate respective tRNA. So this is the first or the initial step, which you can easily see over here. So this is your, this is your, my dear students, amino acylated tRNA or charged tRNA. Next question. This is multiple time repeated. Many ribosomes may associate with a single mRNA to form multiple copies of a polypeptide simultaneously. Such a string of ribosome is termed as, it is termed as polysome or polyribosome. So you can see there is a single mRNA and there are multiple ribosomes. So this is a single mRNA, and there are multiple ribosomes attached to it, making multiple copies of polypeptide chain. So this is how a polysome or a polyribosome look like. Excellent. Next question. This was asked in NEET 2019. Match the following genes of the lac operon with the respective products. Come on, give it a try, my kids. Everybody in the class, give it a try. Everybody present in the class, give it a try. So in a lac operon, I gene codes for a repressor mRNA that forms a repressor protein. I gene codes for a repressor protein. Z gene codes for beta galactosidase. A gene codes for transacetylase and Y codes for permease. Correct? Any doubts in this? Pretty simple. NEET 2018. Select the correct match. Select the correct match. Give it a try, kids. Give it a try. Iliac Zafri. He actually discovered or worked on DNA fingerprinting procedure. So this is wrong. Streptococcus pneumoniae, Griffith worked on it. So this is wrong. Alfred Hirsch and Martha Chase worked on TMV. No, they worked on bacteriophages. Michelson and Stahl worked on Pisum sativum. No, they worked on E. coli. Francis Jacob and Monod worked on lac operon. That's absolutely correct. Next question. Which of the following is not a part of an operon? So, which of the following is not a part of an operon? Just take an example of a lac operon. You need to imagine a lac operon. So, in lac operon, which of the following is something that you did not see? So, which of the following is not a part of lac operon? Come on, kids, give it a try. Which of the following is not a part of a lac operon? Which of the following is not a part of a lac operon? Come on. Which of the following is not a part of lac operon? Come on. Which of the following is not a part of lac operon? Come on. That's absolutely correct. You will not see enhancer in that. Next question. This was asked in 2019. Expressed sequence tags EST refers to. Expressed sequence tags EST refers to. Come on. Expressed sequence tags EST refers to. It refers to the genes that are expressed as RNA. Next. Identify the correct order of organization of genetic material from largest to smallest. Any guesses on this? Nikhil, Ruxana, Karthik, Savita. Any guesses on this, my lovely kids? Yeah, that's absolutely correct. The largest one is the genome, right? Made up of chromosome, a haploid set of chromosome. Chromosome will have genes and genes will be made up of nucleotides. Simple. Largest, smallest. Next question. This was asked in 2015. Satellite DNA is important because, what is the characteristic of satellite DNA because of which it is useful in DNA fingerprinting? Ma'am, genome का नहीं पता. Genome क्या है? Genome is haploid set, right? अगर आपके in your DNA, my dear students, the total amount of DNA is 2C, right? So if you reduce it, or maybe if you say haploid set of chromosome also, if you want to imagine like that. So in a diploid cell there is 2n amount of DNA and 2n number of chromosome and 2C amount of DNA. So if you reduce it, right, it becomes a haploid cell. So in a haploid cell there is a genome, the DNA is half, amount of the DNA is half, number of chromosome is half. So there is C amount of DNA and n number of chromosome in a haploid cell. Is this clear? Genome is n, half, one set of chromosome. So in a diploid cell there are two sets of chromosome. This is okay, Nikhil? Are you able to relate it? Okay. So the answer is D. It shows high degree of polymorphism in a population and also the same degree of polymorphism in an individual, which is heritable from parents to children. This is the last question, my kids, and I want everybody to give it a try. This was asked in Karnataka NEET 2013. One of the most frequently used techniques in DNA fingerprinting is, come on, I want everybody to give it a try. One of the most frequently used techniques in DNA fingerprinting is, perfect, it is VNTR, variable number tandem repeats. So these were the important questions, my kids, I wanted to discuss in this class. So till now, if you haven't joined to Infinity Learn Telegram channel, do join it to get the updates for the coming session as well as for the PDF of this class. Okay, so till now, if you haven't subscribed to Infinity Learn, my dear students, do consider subscribing to Infinity Learn and do like, share and subscribe to our channel. Thank you very much for listening, my dear students. If we talk about the most important topics, if I talk about in this chapter, so the most important, this is a very lengthy chapter, so the most important topics are the DNA, including the structure of polynucleotide chain and the packaging. Then it is the transcription, which is very important, regulation of gene expression, and then the genetic code. These are the most important topics from which maximum questions has been asked. So please remember this. Okay, thank you very much, Nikhil, Karthik, Mohit, Savita. Thank you very much for joining this session, my dear students. Thank you very, very much. That's all from this class. Bye-bye.